Okay, so first, thank to the Matus for this opportunity to show part of my research. So my talk is going to be focused on biomaterials and cells, friends or enemies. So what I'm going to do is try to show you if we can reach this friendship between biomaterials and cells. So why is that important? That's important because tissue engineering, so the basis is the one that is shown on the figure. So basically we have a tissue that is damaged and needs to be replaced. So the idea of tissue engineering, just take the cells from the patient, from the own patient, just seed them, culture them, expand them. At some point, find the ideal biomaterial to seed them and grow them of them. And once they are already grown and we have the tissue, just go back to the patient once the tissue is complete. So what this means that biomaterials and cells are doomed to be understood. So if we want to ensure success on tissue engineering applications, we need to understand these interactions and to reach a friendship between them. So I'm going to talk to you a little bit about this research line where I'm working with. And so what is ideal biomaterials? So ideal biomaterials are the ones that we're trying to develop on our labs. And they should be biocompatible, hemocompatible, biodegradable, suitable with mechanical properties, enhance cell attachment, promote regenerative process, and adequate for drug delivery. So what I'm going to talk to you about, it's a little bit of how we test each of these points and kind of understand which is a good biomaterial or a bad biomaterial for each of these. So we start with biocompatibility. So this is an example. So we meant to develop coating materials for metal implants using a new glycolipids, which are called sulforolipids. So we had a bunch of molecules with different structures, and the initial test was, okay, so which one should be the one? So what we did is we culture our cells, in this case macrophages, with the different molecules. And what we saw, it's, even though they look quite similar, it's just one is an axial chain and the other one is a pentyl chain. So it's not such a big change from the chemical point of view. What we saw is that one of them, they actually was lethal at high concentration. So from this, we could select our initial starting point. Okay, another property that needs to be tested is hemocompatibility. So in this case, I'm showing you data about ceramic scaffolds that were performed with variable chemical and physicochemical properties. So the initial phase of the study was to try to understand how is the network, our ceramic network. So we assess the density, the surface area, and the porosity of all the ceramics. And as you can see, they are quite close between them. And then we check the hemolysis. So we culture, we test our samples with a whole blood cell, and we check how much hemolysis was caused. So what we saw, it's a high amount of hemolysis for one of the samples. And actually, it is really high because the hemolysis limit that should not be increases 5%. So we have the sample with really high amount. And so what we did is we went back to the lab and said, okay, how can we explain this whereas there is no such a big change in physicochemical properties? And then what we analyzed was the contact angle, so the hydrophilicity, so the interaction between the biomaterial and the water. And what we saw is that actually was quite different between the ones that are here, which is silicon carbide ceramic versus carbon ceramic, which was clearly more hydrophobic. So at this point, this material was stopped to work with. So next step that's also really important for tissue engineering is to be biodegradable. So to make sure that you allow the new tissue to be formed when the biomaterial is degraded. So in this case, I show you an in vivo assessment of the regenerative capacity of polymeric scaffolds They are chitosan foams with microspheres that have been loaded with either BMP2, which is bone morphogenic protein 2, which is going to promote formation of bone, alendronate, which is known to decrease bone resorption, so increase bone formation, and MMP9, which is a enzyme that is going to promote the bone remodeling, so the increase in bone formation. So what I show you here, it's the control group, where we see here this Calvaria critical size defect. So we show the scaffolds at 6 and 12 weeks. And when we analyze the bone repair, what we saw is that the ones that should have higher bone repair, which are the ones with MMP and BMP2, they were not actually giving such a high effect. Why? It's because when we check the degradation rates, what we saw is that these scaffolds were not degraded as fast. So we are promoting the formation of bone, but the scaffold is not getting degraded. So there is no bone that can be produced. So this is the point that we found. So another point that it's actually really important is to ensure that you allow cell attachment. So if we have a biomaterial that is going to be seated with cells, we need to make sure that actually cells attach to the biomaterial, because if not, it doesn't make much more sense. So in this study, what we did is we tried to increase the endothelialization of vascular grafts. So vascular grafts to be functional and to inhibit coagulation and thrombus formation, it's needed to have an endothelial layer that is going to secrete molecules to the platelets to say, okay, don't attach, this is not a harm material. So what we did is we just work with the plain biomaterial, which is PLA PhD, and we seed the cells. So what we saw once we seed the cells is that they were not cell attachment at all. So what did we do to improve this? So what we did is to include heparin, which is a molecule that is known to control endothelial cells homeostasis, and to the surface of the scaffolds. So what we saw is indeed an enhancement in cell attachment. And here in the confocal microscopy, you can see the clear difference between the control and the functionalized scaffolds. So the green dots that you see here are the cells, and the blue ones are just actually, it's autoreflections from the biomaterial. Okay, another point, it's mechanical properties. So it's actually something that we already talked about. So mechanical properties need to be the one suitable for the tissue. So here I'm just showing you an example of small diameter vascular grafts that were performed with variable compliance. So which means variable compliance means that they behave differently with the pressures. Okay, so why is this important? This is important especially in small diameter vascular grafts, because when we work with biomaterials that have low compliance, every time the heart is beating, the diameter of the vessel, of the native vessel, is going to increase, but not of the scaffold. And so what happens in this case, that the shear stress is really high in the suture line between the artery and the graft. So the cells are subjected to high stress and they start to increase the thickness of the intima. And at some point, usually the vein collapse and the graft needs to be replaced. So this ideally will not happen if we have high compliance graft. That would mean that it will behave as the native artery. So when the heart beats, the diameter increase. So in order to actually check this mechanical behavior, what we did is we set up a bioreactor system in our lab. And so we have the media flowing. Here we have the graft and the arteries that were using porcine arteries. And then we just pump the flow. So hopefully this is going to work. Okay. No. Yes, but I'm clicking, it's not going, I don't know. Oh, no. Okay, yes, now it's going. So here you see the one with high compliance. So as the higher speeding is clearly the change in the mechanical properties. Here is the low compliance, and here is the control, just the pure PTFE, which is the one that has been used in clinic right now. Okay, so once we set up our culture, what we did is we actually test the behavior of the tissue surrounding the suture point between the graft and the vein. So here I show you the immunohistochemical staining using 2 markers of smooth muscle cells. So smooth muscle cells are supposed to be in the media of the vein, or in the intima, so in the medium side of the vein. And so they are the ones responsible for contracting. So these are 2 of the proteins, which is smooth muscle alpha actin and smooth muscle 22 alpha. And so as you can see in the sham control, which is the vein itself, there is homogeneous staining on them. While when we work with PTFE, we saw an enhancement close to the intima, so the blood is flowing this side. So this will be the intima of the vessel. And in high compliance, we saw that's kind of the similar pattern as the sham control. So indeed, improving the mechanical properties gave us better data in terms of tissue structure and tissue anatomy. So another of the points that are gaining a lot of attention in the tissue engineering field is to have a biomaterial itself that is able to promote the regenerative process, either alone, just by their surface properties or network structure, or when we include different molecules, that usually growth factors or synthetic molecules, to promote this specific regenerative process. So here I'm showing an example. This is alginate hydrogels that are quite commonly known for being really highly inert materials. And so what we did is we increased the bioactivity of these hydrogels by adding calcium carbonate. So calcium carbonate from marine origins. So I show you here 2 scanning electron microscopies of the different particles of calcium carbonate that were added. And as we saw, where we checked the relative ALP activity, which is a phosphatase activity directly related to the osteogenic differentiation of mesenchymal stem cells, we saw that indeed, when we add, especially in the case of oyster calcium carbonate, we saw a clearly increase in the alkaline phosphatase activity. Was lower than with hydroxyapatite, which is the gold standard ceramic for bone regeneration. And we also test the glycerin red staining, which is going to also give us information about the formation of calcium deposits, which is directly related to osteogenic differentiation. We also saw the same trend. So ideally, the last point that I just mentioned on the initial slide is that this biomaterial should also be adequate for drug delivery. And we have worked with a lot of mechanisms for drug loading and release. So really, in a summary way, we can have unspecific absorption. We can load biomaterials with unspecific absorption, physical interactions, physical entrapment, or covalent union. And the release profiles are going to be conditioned by the loading technique used. So this is just a fast sample. So this is a hydrogel meant for intraarticular administration and treatment of osteoarthritis, which is known to have a really high levels of inflammatory cytokines. So we load it with beta-lactam, which is an anti-inflammatory drug. Initially, we test the drug release profile, which was a linear release profile characterized by a diffusion control release mechanism. And then we tested our hydrogel on a synovial cartilage explant co-culture. So We take cartilage and synovium from osteoarthritic patients. We extract them, cut them, and co-culture them. So once we have our co-culture established, we check the activity of dexamethasone, which is the molecule that has been approved in clinic. It's actually being used for this treatment. Then the drug alone, and then the drug on our system. And we saw that actually our system was able to decrease the IL-8, which is a pro-inflammatory cytokine. So this is a really brief summary of all the techniques that I just mentioned. We have a lot of more of them that are available in Imatus. I'm also open for any type of collaboration that you may think of. Just to find, thanks for the funding bodies to allow me to do all this research that I just showed you. Thank you for your attention, and I'm open for questions.
Questions, comments, ideas? Yeah, I do have one. Regarding these systems that show different compliance depending on flow or pressure or so, are they made by electrospinning?
Yes,
or?
they are electrospine mesh. So what we did is in order to be able to change just the mechanical properties and not also the chemical properties, we just changed the thickness of the electrospine mesh that we used.
Oh, okay, it was a question of the resistance, mechanical resistance of the external layer just by adding more amount of polymer.
Yep.
Okay. Well, if any of you is interested in the cell cultures or this kind of compatibility or differentiation tests, I am pretty sure that Patricia is very open to any collaboration with all of you. Okay, therefore take in mind. No comments, no more questions. Okay, let's thanks again, Patricia.